Welcome to The Explainer. I am so pumped to have you here today. We are jumping straight into a monumental, heavyweight matchup between the two absolute titans of third-generation sequencing, Pacific Biosciences and Oxford Nanopore technologies. Today, we're decoding exactly how these two wildly different platforms are fundamentally revolutionizing modern genomics. Short reads changed sequencing, but long reads complete the picture. To really get why this matters, think of your genome like a massive jigsaw puzzle. Next-generation sequencing, the short-read stuff, is basically like trying to put together a 10,000-piece puzzle of a clear blue sky. You've got millions of tiny 150 base pair pieces, and figuring out where they overlap is a nightmare, especially in highly repetitive areas. But long reads, they give us massive continuous chunks. Suddenly, that impossible blue sky puzzle turns into just a few large pieces that snap together effortlessly. Here's our fast-paced roadmap for today. We'll cover the long-read revolution, dive into PacBio's optical sequencing, thread the needle with Nanopore's electrical approach, put them head-to-head, explore real-world applications, and wrap up with the future of genomics. Let's hit it. Section 1, the long-read revolution, from Sanger to third generation. To appreciate this tech, we have to look back. In the 1970s, we had first-generation Sanger sequencing, the foundational method that actually gave us the Human Genome Project. Then, the 2000s brought next-gen sequencing, with platforms like Illumina. Incredible depth, way lower costs, but it relied on PCR amplification and those tiny, fragmented short reads. Fast forward to the 2010s, and the third generation arrives. The absolute game-changer here? Methodologies like PacBio and Oxford Nanopore sequence single molecules in real time with the zero PCR amplification. That means no amplification bias, and the birth of the true long read. Section 2, PacBio and sequencing by synthesis. This one is all about optical precision. Let's look at our first contender, Pacific Biosciences, or PacBio. Their approach is called single molecule real-time, or SMRT sequencing. And the real magic happens inside these microscopic chambers called zero-mode waveguides, or ZMWs. Picture millions of these tiny little wells on a silicon chip. Inside every single one, there's a single DNA polymerase enzyme anchored right to the bottom. As a strand of DNA gets pulled through, that enzyme builds a matching strand using fluorescently labeled nucleotides. Every time a base gets added, bam, it emits a distinct flash of colored light. It's literally recording a high-speed, cinematic movie of DNA synthesis in real time. It's wild. And because PacBio reads that same circular piece of DNA multiple times, it generates these super accurate consensus sequences called hi-fi reads. When it comes to taxonomic profiling performance, PacBio's hi-fi metagenomics absolutely obliterates traditional short-read methods in precision and recall. We are talking Q30 accuracy, meaning it's 99.9% accurate. If you need flawless reference-grade data, this is the gold standard. Moving right along to Section 3. Nanopore, threading the needle. Let's talk electrical innovation. Enter contender number 2, Oxford Nanopore Technologies, or ONT. They take a totally different, almost sci-fi approach. No optics, no flashing lights. It's all about electrical current. Imagine a specialized protein pore, a nanopore, embedded in an electrically resistant membrane. A motor protein literally threads a single strand of DNA or RNA straight through that pore. As the strand squeezes through, it disrupts the ionic current flowing across the membrane. But it doesn't just read one base at a time. It reads short sequences of about 5 bases, known as k-mers. From there, heavy-duty neural networks decode those electrical disruptions in real time to tell you the exact sequence. Now get a load of this scale. Because Oxford Nanopore doesn't rely on polymerase synthesis in the same way, your read length is basically only limited by how intact you can keep your DNA sample. They can achieve ultra-long reads of over 4 million base pairs in a single unbroken strand. That is just incredible for bridging massive structural variants or those super tricky repetitive regions of the genome. Section 4, the head-to-head comparison. Let's put optical and electrical in the ring together. In the optical corner, we've got PacBio. It uses fluorescence pulses, it requires some heavy centralized lab machinery, but it delivers that jaw-dropping Q30+ accuracy. In the electrical corner, Oxford Nanopore. It reads ionic current disruptions, it's famous for pocket-sized portability, literally devices you can plug into a laptop, and it is the undisputed king of ultra-long reads. Let's break down the hard data on length, accuracy, portability, and turnaround. If your absolute top priority is read length, ONT gives you up to 1 megabase and beyond, whereas PacBio Hi-Fi hovers around 15 to 30 kilobases. However, for pure accuracy, PacBio is currently superior with its Q20 to Q30 plus reads, though ONT is catching up fast. Portability? ONT has the minion, PacBio doesn't. Turnaround time? ONT streams data in real time, in minutes, while PacBio is fast but operates in a more traditional run cycle. This quote from a bioinformatics expert perfectly captures the industry vibe right now. They say PacBio's accuracy is still the gold standard, but ONT is easier and quicker. It really comes down to what you value more for your specific project: the absolute confidence of PacBio for resolving complex human alleles, or the pure convenience and speed of Nanopore. Section 5. Real-world genomic applications. Where do we actually deploy these things? The truth is, there isn't a single universal winner. The choice entirely depends on the application. You deploy PacBio when you're building flawless telomere-to-telomere genome assemblies, or when you need high-accuracy structural variant calling. On the flip side, you grab Oxford Nanopore for field pathogen diagnostics, like the Lampore assay from the COVID era, or any rapid, portable clinical testing where you need to bring the sequencer to the sample rather than the sample to the lab. Let's quickly see how PacBio handles transcriptomics with its iso-seq workflow. First, you extract the total RNA, synthesize the first strand cDNA, do some PCR amplification, and finally construct a SMARTBell library. By following these steps, PacBio can sequence full-length transcripts up to 10 kilobases. This is huge because it captures the true start and end sites of transcription, totally bypassing those annoying truncation issues you get with other methods. But then we look at ONT's minion, and it opens up a totally different, highly specialized world. Real-time, pocket-sized pathogen detection directly in the field. This thing is the size of a candy bar. Researchers have literally taken the minion into the middle of rainforests for biodiversity assessments and right into the epicenters of viral outbreaks for real-time tracking. On-the-spot diagnostics without needing a massive lab setup. Simply amazing. Finally, Section 6. The future of genomics. Reading deep. So, where is all this heading? The massive adoption of third-generation sequencing is exploding, driven by huge global investments. Pathogen detection is leading the charge at 40%, fueled by our need for rapid outbreak responses. Right on its heels is precision medicine at 35%, and rare disease research at 25%. These long reads are finally letting us spot the incredibly complex structural variants that short-read tech completely missed for decades. Which leaves us with this one big question. We're entering an era where long reads don't just read long, they read deep. As these sequencers get smaller, faster, and even more accurate, how will real-time portable sequencing change the future of your personal medicine? Are we going to see rapid genomic diagnostics sitting right there in your primary care doctor's office? Honestly, the technology's already here. Thank you so much for joining me on this explainer. Keep wondering, keep exploring, and I'll catch you next time.